Hello and welcome back to Survey of Biology, and we're in Unit 9, Section 3, where we're going to start talking about meiosis. If you remember, Units 1 and 2, it talked about the cell cycle, the entire life cycle of a cell from interphase through mitosis to get two identical daughter cells, and then we talked specifically about mitosis. And that's what happens as somatic cells or body cells, and we end up with two identical daughter cells. Now we're going to take a look at what happens in meiosis. So this is the production of gametes, or sex cells. So this will be the sperm and the egg. And the big thing to remember about meiosis is it's something called a reduction division. So what we're going to do here is we're going to take the chromosome number from our normal 46 for humans, which is called diploid, and we're going to reduce it down to half that number, 23, which is haploid. So meiosis is going to produce gametes which are haploid in their chromosome number, and for humans that number would be 23. Okay? And one of the big differences between mitosis and meiosis is mitosis, if you remember, produced two identical daughter cells. Meiosis is going to produce four genetically different daughter cells. So here's where the genetic diversity comes in that we see in the offspring of people. And there's three ways that it increases diversity. The first is something called crossing over, and that occurs in P1 or prophase 1 of meiosis. There's also something called independent assortment that we'll talk about that occurs in metaphase 1. And then finally there's random fertilization. So the sperm or egg produced, it's random which one actually results in an egg or a zygote. So let's look at the steps of meiosis. So the first thing I want to do is let's make sure that we understand the difference between haploid and diploid. Most of the cells in our body, our skin cells, nerve cells that are functioning, are diploid. They have two copies of the genetic material, and those are what we call, if you remember, the somatic or body cells. Now, where do those two copies come from? One set would be maternal from mom, and one set would be paternal from dad. So if we look here, we have three pairs of what they call homologous chromosomes. So we've got the red, one would be from mom, one would be from dad. We've got the blue, again, maternal, paternal, and then we've got the green. Now, if we look at just the green ones here, these are called homologous chromosomes because they're going to code for the same genetic information. Now, what happens in meiosis is the maternal and paternal DNA gets separated, and we end up with half the normal amount of DNA, and we end up with what we call a haploid cell over here. So you can see here we've only got one copy of green, one copy of blue, one copy of red. So we no longer have the homologous pairs, we just have one set, and that's what produces the gametes, the sperm and the egg that we're going to see. So meiosis makes four haploid daughter cells as the result. So if we look at meiosis, it can be broken down into two parts. There's meiosis 1 and meiosis 2. This section is just going to look at meiosis 1. And the big thing here is the chromosome gets reduced by 1/2. This is what's known as the reduction division. So in the resting state of a cell, we've got our normal chromosome number. For humans, it would be 46. We go through the cell cycle, so DNA replication happens during the S phase. And then what happens is we lead into, similar to the phases of mitosis, prophase, metaphase, anaphase, and telophase. But in meiosis, what happens is the chromosomes will condense, and we get what are called homologous chromosome pairs here. So we've got a copy. If you look at the orange ones, those might be maternal, so there's two copies from mom. And then the striped ones would be paternal from dad. And they line up. Something called crossing over can occur that increases diversity. Those then line up on the metaphase plate, and then they get pulled apart during anaphase, and then telophase pinches them off, and halfway through we get two new cells. But remember, we're only halfway through meiosis. There's one more division that will take place. So now let's take a look at each of these steps individually. Excuse me, we got one more kind of big picture in a cartoon version of it. And again, you can see we have interphase. So during interphase, the chromosomes would replicate during the S phase. We then have prophase, where the DNA will condense, And here's where we get what we call those homologous chromosomes, or a tetrad, where we have actually 4 copies lined up. Then they go to metaphase, where they'll line up along the metaphase plate right down the middle and then get pulled apart. So this is where something called independent assortment can take place. And finally, during the anaphase and telophase, they're pulled apart, and we're halfway through with 2 cells. Now, let's make sure that we understand this concept of homologous chromosomes and something called sister chromatids. Okay? So homologous chromosomes are going to be chromosomes that will carry genes controlling the same inherited characteristics. So those are going to be homologous chromosomes. So if we look in a normal cell, you'd have 23 pairs, and each pair would be paternal, the orange in this case, and maternal, the red. So you would have 2 genes that would code for the same traits. So maybe eye color might be on here. And maybe you have a gene for blue eyes from dad, and maybe you have a gene for brown eyes from mom. They're homologous chromosomes because they carry the same inherited characteristics. Now, as we get ready for division, during the S phase, we would duplicate that DNA. So we'll get a copy of the paternal right there. Now, those 2 copies are called sister chromatids. And the same thing happens with mom's DNA. We get a copy here, and this X, those are sister chromatids. If we put those 2 pairs of sister chromatids together, those are the homologous chromosomes. That's a homologous pair, and sometimes because there's actually 4 copies, that structure is called a tetrad. And here's what it looks like here. So remember we talked about the centromere, the kinetochore during mitosis, about where the spindle fibers will attach? Well, if we look at the blue chromosome there, the blue is 2 sister chromatids of the paternal, dad's DNA. The red would be 2 sister chromatids of the maternal, mom's DNA, and all together they make homologous chromosomes. So this is with the tetrad. So we're going to talk about 2 divisions where first the homologous chromosomes get separated, and then second, during meiosis II, the sister chromatids will get separated. So prophase 1 is the first step. And if we look at 1, the thing here that happens is chromosomes will coil, they'll condense. The homologous pairs recognize each other and align. That's called synapse. That's where we get the tetrad between those homologous chromosomes. And it's here that crossing over at the chiasmata, that's kind of a funky word that means the position where crossing over will take place. But crossing over is going to give us genetic recombination. So if we go back to the orange and the red, orange being dad's DNA and the red being mom, so we've got our homologous chromosomes, or tetrad, right there. And they'll line up, and right at this location right there, the chiasmata, what will happen is little pieces of the DNA will flip-flop. So all of a sudden, if you look down here, we've got combinations of DNA, combinations of genetic material that weren't together before. So this is why you really are a combination of your mother and father, but it might not be all the same traits, because this is where genetic diversity gets increased. So again, here's another picture crossing over. So we've got our homologous chromosomes. We'll get the chiasmata right there where they cross over, and now we get new combinations. So if you look here, we have a lowercase c, which maybe is color of your hair, with uppercase A and B, and vice versa over on the red one. So that's where prophase 1 is where crossing over will occur. From prophase, we head to metaphase, and here's where the different homologous chromosomes will line up. So the nucleolus and the nuclear membrane disperse. The spindle fibers will attach to the kinetochore, so remember we talked about the centromere in the middle, and the tetrads, the homologous chromosomes, line up at the metaphase plate. The homologs or homologous chromosomes are on opposite side. It's here where independent assortment can take place because it's random as to if the maternal or paternal DNA ends up on one side or the other. So if we look at two sets of chromosomes, so two here, blue is dad, red is mom, and there's two possibilities. We can either have all blue on one side, or we can flip-flop over here on the right. We can have red and blue switched up. And the big thing to remember, each of those will result in a different combination. So if there's only two chromosomes, there's 4 different possible combinations that we can end up with. If we have 3 chromosomes, like in this picture here, there's 8 possibilities because of independent assortment. So independent assortment increases diversity. And from metaphase, we head to anaphase. So here the kinetochores do not split. The sister chromatids remain intact. So this is very, very important. So what happens during anaphase 1 is the homologous chromosomes are what separate. They get pulled apart. And the homologous chromosomes will separate, I'm going to add randomly. So that's the independent assortment piece. And here's the reduction division. Here's the important one where we go from being diploid to getting those haploid cells that we classify as only having half the number of chromosomes. So meiosis 1, anaphase, pulls apart the homologous chromosomes. The sister chromatids stay together. They're being pulled to the opposite pole so that we can start to pinch them off to get 2 new cells. And again, here's with 3 chromosomes. So the sister chromatids, all of these stay intact, and it's the homologous chromosomes that get pulled apart as we've transitioned from anaphase into telophase. So telophase is where the cleavage furrow starts, and if we think about a cleavage furrow, what is it? Basically, it's a little dent in the cells. Think back to mitosis where it starts to pinch off. And sometimes after it pinches off, we may get what's called interkinesis. And the big thing to remember here is DNA does not replicate. So at this point, we've got 2 cells, and we're ready to head to meiosis 2, which will be the subject of our next section. And meiosis 2 is a little bit easier. It's similar to mitosis. But let's go back and look at the end of interkinesis, what we actually have. So telophase, we see that there's a pinching in. The chromosomes are starting to recondense. The nucleus may reform. So the chromosomes are at the pole and are becoming more diffuse. The nuclear envelope is reforming, and the cytoplasm may be dividing as it goes to telophase. And then finally we have interkinesis. And the big thing to remember here for interkinesis is that we end up with haploid daughter cells. So we end up with 2 of them, okay? So there's no further replication of the DNA. And the other big thing to remember here is we're only halfway through. So there's one more step, but halfway through, we've got 2 daughter cells that have half the amount of DNA, and they're genetically different from the parent cells. So again, thank you for your time, and we'll see you shortly as we continue with the second part of meiosis.